In 1958, two young scientists settled one of biology's deepest questions: how a living thing copies its own DNA without ever watching it happen. They answered it by weighing molecules. It has been called the most beautiful experiment in biology. 5 years earlier, Watson and Crick had found the shape of DNA. The double helix, two strands twined together, each the mirror of the other. And they noticed something. If the strands came apart, each could template a new partner, one molecule becoming two faithful copies. A thrilling idea. But noticing a possibility is not proving it. And by 1958, no one had watched DNA copy or knew what became of those two original strands. 3 answers competed. Semi-conservative, the helix unzips, and each copy keeps one old strand and one new. Conservative, the original helix stays whole, and a wholly new one is built alongside it. Dispersive, the old DNA is broken up and scattered, old and new interleaved along both copies. 3 pictures of a single event, and to tell them apart was to learn how inheritance is passed on. Matthew Meselson and Franklin-Stahl saw that the 3 pictures disagree about one thing: where the old atoms end up. So they made old and new DNA weigh different amounts. They grew bacteria for generations on heavy nitrogen, nitrogen-15, until every strand was dense. Then, in a single moment, they switched the cells to ordinary, light nitrogen-14. From then on, every new strand was light, and every old strand stayed heavy. But how do you weigh something a hair heavier than its twin? With a method Meselson built with Jerome Vinograd, spin the DNA in a cesium salt solution at tremendous speed. The spin packs the salt into a density gradient, light at the top, dense below, and every molecule drifts to the one layer that matches its own weight. Heavy DNA bands low, light DNA high, and half-heavy DNA settles exactly between. A photograph in ultraviolet light makes the bands appear. At the start, every molecule was heavy, one band, low in the tube. They let the cells divide just once in light nitrogen and spun again. The heavy band was gone, replaced by a single band at the halfway mark. Every molecule now exactly half heavy. That picture killed the conservative model outright. Had the old helix stayed whole, there would have been 2 bands, one heavy, one light. There was one in the middle. But a single halfway band still fit 2 models, semi-conservative and dispersive, both predicted after 1 round. So they let the cells divide once more. After the 2nd generation, the band split. Half still at the halfway mark, half now fully light. That removed the last rival. Had the old DNA been chopped and scattered, the band would have drifted lighter as one piece, never splitting cleanly into a hybrid and a light band. Every generation fit the same march: heavy, then all half-heavy, then half-heavy and light in equal measure. And for proof, they heated the halfway DNA until its 2 strands parted, and they came apart as one heavy strand and one light. The hybrid was exactly what semi-conservative replication demanded: one old strand paired with one new. That is semi-conservative replication, and it is how almost all life copies its DNA. Each time a cell divides, the helix unzips, and every strand builds a fresh partner. So each new molecule carries one strand made today and one inherited from the past. Watson and Crick's quiet guess had been exactly right. Its beauty is its economy. 3 sweeping ideas about the nature of inheritance and one clean measurement told them apart, leaving nothing to argue. No tracer to interpret, no model to assume, only heavy against light, and bands in a tube you could photograph. It earned the name, the most beautiful experiment in biology. Sometimes you don't need a sharper lens, only a cleverer question. Beautiful experiments